First of all, thank you so much for the invitation. I'm glad that Luisa was able to present as well. So this will be like a segue to what she presented. And this is more towards like the development of therapy. And we discussed with the foundation before that maybe the best way to go for this condition would be like a gene therapy. Gene therapy, as you know, we are targeting the root causes of the condition. So we are there to replace, not replace, but to add the right copy of the gene, the right version back into the brain cells, hoping they would work properly. But how to test that? So that's a big question, and we need like a good model, and the brain organoid. It is the model of choice, and more people are recognizing that this system, even though it's very reductionist, is not an organism, but it can help us to give the proof of concept, which is the first stage to actually convince ourselves that this strategy might be good. And I didn't invent brain organoids. Actually, who invented was Yoshiki Sasai from Japan in 2008. So he was really the one who showed us that you can take human pluripotent brain cells, human pluripotent stem cells, grow them in suspension, and he noticed that the cells will self-assemble into a tissue that resemble the human brain. And we have some pictures here, which is quite remarkable because it is very similar to the human developing fetal brain. An anatomist will probably not distinguish between those two, except for the lack of vascularization. This is something that the model is still incomplete, but nonetheless, we are working on that. So hopefully the newest version of brain organoids will contain vascularization as well. So how to turn this technology into something that is useful for the families. And before I continue, let me just highlight that you're going to read in the news that this technology is called like a mini brain in a dish. And that's okay. It's a layaway to convey the idea that what we have is a brain tissue, and people from the outside of science might understand. But I want you to understand that this is not exactly what happens. We don't have a miniaturized version of the human brain in a dish. Instead, we have like a reductionist model that has intrinsic limitation as any other model. Even the animal models have limitations. And what are those limitations? Well, I mean, as I mentioned, this represents more on the fetal stage of the brain. We are not recapitulating, we are not recreating brain cells that resemble like an adult brain. We are in an early stage, so this would be more equivalent of a fetal brain. As I mentioned, they are not vascularized. We are learning how to vascularize. And because they're not vascularized, the size of these organoids are limited. And I'll mention in a moment what are the sizes and why they don't grow bigger. So we don't have all the cell types represented. The human brain contains like 100 billions of cells. There are more or less 200,000 of different subtypes of neurons. We are not even close to capture that diversity. We don't even know if we are growing them in the same conditions, in the right conditions that happens in the human brain. So there is lots of unknowns, and because of that, these are like limitations that we need to keep in mind when we use that model. And nonetheless, I'll mention that also as we increase the complexity of this model, ethical and moral questions will arise. And why I'm saying that? I'm saying that because someone who doesn't understand the purpose of what we are doing might want to halt the science because of ethical and moral questions. But the more people understand the purpose of this model, the better, because then we can argument that the model is useful for treat neurological diseases. We're not just trying to play God here and recreate the human brain for no purpose. There is a real purpose on doing that. So, okay, now that we understand the limitations, what my lab has been doing for the past several years was to optimize a protocol that makes these brain organoids more reproducible and better, including having different cell types that the previous Sasai protocol didn't have. And we recently published a very detailed protocol on how to reproduce that. And now anybody in the world could take our methods and do it in their labs. And we call that a semi-guided cortical organoid. So it is the cortex, but we are assimilating cells that are coming from the outside of the cortex, for example, inhibitory neurons. And it always starts with human pluripotent stem cells that we dissociate, and then we neuralize them using the dual SMAD inhibitors. These are two factors, two protein factors that we add in the culture. One of these factors will inhibit the stem cells to become any other tissue in the body but the brain. The other factor will stimulate the brain cells to proliferate. That's exactly what we want. So by blocking all the other cells and by stimulating the brain cells, we hope that this will induce the proliferation of neuroprogenitor cells. So these are the cells that will give rise to all the brain cells in the brain. So this neuroproliferation happens in two sequential stages where we add growth factors to stimulate them to grow. These are FGF and EGF. And after that, we let the cells to differentiate or to specialize. And after that, they will mature and continue to mature until they start forming synapses, forming connections, and creating networks inside of these spheres. So this is how we grow them in the lab. So we grow them on these like 6 well dishes, and each one of these white dots is the beginning of one brain organoid, and they can reach 0.5 centimeter in diameter with roughly a couple million cells, more or less 2.5 million neurons in there. And they don't grow bigger, again, because they are not vascularized. So the inside of the cells do not receive the proper nutrients to continue to proliferate. So they stop proliferating. Eventually, the center will become necrotic. But the surrounding cells, the active neurons, would continue to be alive. And honestly, they would be alive as long as you keep feeding them for several years, if not for the same time as the brains in our the cells in our brains for a human lifetime. So we just don't know how long they last because nobody in the lab will keep keeping them going for several years. So we have other better things to do. So how they form, how the brains form. So this is like a two-dimensional representation of one of these brain organoids. Imagine that the top of the slide here contains the center of the organoid where the progenitor cells are located. And I hope you can see that there are cells that are migrating from inside to the outside. So that's exactly how the brains form, from inside out. And they take advantage of some of these cables. These are radioglia cells that happens to be there and to guide the progenitor cells to migrate from inside out. As soon as they reach the outside, the external part of the organoid, they start to differentiate and form connections, and that's how the first neural networks start to be formed. So if you slice one of these spheres, what you see is that the center of the spheres will contain a ventricular zone. It's sometimes more than one, most of the time more than one, surrounded by progenitor cells. These are the cells that proliferate, as I mentioned, and they will migrate out following these cables here. These are the radioglia cells, the process from these radioglia cells, and they start to differentiate and form these layers. The layer formation is what builds all the network inside the human cortex. So this is the precursor of all the cognition. This is the precursor of all your motor function, your visual cortex. It's all there. It's all encoded genetically in these cells. And the way the cells self-organize represents how the adult brain will actually function. So you can imagine if there are defects during this process, the adult brain will suffer, will have like a dysfunction caused by either the migration of the cells or in a DLG4 condition, we think that is about the communication of these cells. And so some people ask me, okay, but what kind of cells you have? So it's a very dynamic process. The type of cells that we have really depends on when you look at the organoids. In the beginning, it's all about progenitor cells, but as they migrate, as they differentiate and mature, more and more diversification of the cells will emerge. So this is a snapshot at 4 months. You can see the pool of progenitor cells remains there. Most of these progenitor cells are specializing into glutamatergic neurons. These are excitatory neurons, the type of neurons that produces excitation. There is some of these progenitor cells that also will become glia cells. Most of the time, these are astrocytes. These are cells that are there to help form the connections. There are also a discrete population here of inhibitory neurons. This is one of the advantage of our protocol compared to others. We do have the inhibitory neurons that emerge in there spontaneously. We don't need to add the inhibitory neurons during the process. They will emerge from our protocol and help to create the what we call the AI balance, excitation and inhibition balance, that dictates lots of neurological conditions such as autism or epilepsy, for example. All right. So one important point for us, especially for conditions where synapses are important, it is if you can measure the electrical activity moving from one neuron to another neuron. And that's what we call the formation of networks. And as I briefly mentioned before, the early stages of these networks is what's going to create circuitries in your brain that are responsible for the cognition or all your functional actions that you have throughout the day. So it's very important for us to actually capture that. And the only way that we found for now to understand the evolution of these networks over time is to place the organoid on top of a dish that contains a multi-electrode array. So in the bottom of the dish, we printed electrodes. And these electrodes, when they are in touch with the organoid, the cells from the organoid, they will capture the electrical activity and they will send us a signal generating this activity map here. So all the electrodes are here. You cannot see them. But when the neurons that are nearby fire, you capture their activity. And you can do that over time. That's the beauty of this technology. So you can actually follow them over time. So what happens to these organoids if we follow them for 1 year? So that's exactly what we did. So we followed the networks for about 1 year. And what we learned is something quite interesting. In the first month, most of the connections that are being formed are a little random. So the neurons are starting to connect to each other. They're forming the synapses. The astrocytes are there to adjust. But the network is not there yet. So they are a little bit random. The networks start to be formed when they are about 4 months of age. And by 6 months, you see large synchronization. So all the population of neurons inside these organoids start to fire in synchrony. And that's what we see here in all these peaks. After that, we start to see a desynchronization or an increase in complexity. So this follows the different frequencies that we can actually capture from an electroencephalogram, an EEG, when you place the electrodes directly into your skull. So we start seeing the alpha, delta, gamma waves that we actually can compare to the EEG of the person to whom we derive the organoids. So that's really cool, even though it takes a while. So that's one of the downsides. The model is so good, but so good that it follows human development, and human development is really slow. We take 9 months to start maturing this network. So in a way, sometimes it becomes impractical because growing the cells for that long, there is an accident or a contamination or something that becomes impractical. That's why we try to avoid these long-term experiments. But nonetheless, having like the initial orientation of the neurons where they start making connections is already powerful enough. And I'll give you an example of one of the first works in the lab where we work with a syndromic form of autism called Rett syndrome. And what we saw in Rett syndrome was something quite interesting. So we're going to see here a video. It's a fly through a network of neurons. The process of the neurons are in green. The synaptic puncta are in red. And I also place in blue the nucleus of the cells, the neurons, so you can actually visualize the size of the puncta compared to the nucleus. So you don't need even to quantify here to realize that the networks of glutamatergic neurons from Rett syndrome are not making as many synapses as the control neurons are doing. And it's not a matter of timing. The Rett syndrome neurons will never catch up. They will never mature. They cannot produce as many synapses as we see in the controls. And that's a very important phenotype. It is so important that we start using that phenotype to screen drugs, to find pharmacological alternatives, and even a gene therapy. So we actually use this model as a proof of principle for a gene therapy for Rett syndrome that it is now in clinical trials. But also important is to find pharmacological interventions. So this is like one example of a drug treatment that we had where we treat the cultures with IGF-1. This is insulin growth factor 1, and it's a molecule that stimulates the production of synapses in your neurons. And so you can clearly see here through the quantification that we increase the number of puncta, synaptic puncta, by just exposing the neurons to IGF-1 for 2 weeks. 2 weeks was enough to make the neurons firing again or forming more connections. And that what is the basis of trofinetide or Daybue, the medication that was recently approved for Rett syndrome. So it shows that the model is powerful enough to produce proof of principles that we can revert the condition, that these conditions are not permanent, that the alterations that we see, even at the synaptic level, are not permanent, but can be reversible. So it's a concept that in neuroscience, at the time, this is 2010, we didn't have. We thought that all these neurological, neurodevelopmental conditions might not have a treatment or a cure because they pass the critical period where the brain might have all the plasticity. But actually, we're showing the opposite, that no, the brain continues to have plasticity as long as you know how to stimulate them. So that's good news for us. But let me go back to DLG4. So what we decided to do is to use an AAV vector. This is an AAV serotype 9, which is the only serotype now that is approved for clinical trials. And when we designed that, we kept in mind that we don't want to reinvent anything novel here. So we want to create a tool that if it works, we can move to a clinical trial really fast because the vector itself is all already approved. There is no new thing that the FDA or any other regulatory agents might see as a problematic. So the only novel piece in this vector, it is the DLG4 gene. So all the rest, all the components, this is the synapsin 1 promoter. It's already approved for FDA. This is the WPRE. So this is like a piece of DNA that helps to stabilize the RNA. So this is all FDA approved. So we're not, again, reinventing the wheel. We are just replacing the correct form of the gene in one of these vectors. Sorry. And so because we don't want to interfere with that, and we want to be ready for a clinical trial, we didn't include any marker. Because if you include a marker, you kill the vector. The vector is no longer clinically oriented. So we decided to create, so we can visualize, create like a sister vector that is virtually identical, but instead of a DLG4, we just put the GFP, the green fluorescent protein. So we can visualize the infection of the neurons in the organoids. And that's what we see here. So this is like under the fluorescent microscope. After the AAV infection, you see many cells that are green. So these cells receive the AAV vector. And if they receive the AAV with the GFP, they're most likely receiving the vector with the DLG4 as well. So it's not a direct evidence, but it's strong enough to convince ourselves that the vector is infecting and is delivering the gene inside the cells, either the GFP or the DLG4 in parallel cultures. All right. So okay, let me go to this slide here. And I believe that this is one of the slides that Luisa presented. So we were able to reprogram cells from several DLG4 mutations. Some are nonsense, frame shift, splice, or missense mutations. And we measured the amount of PSD95 puncta in these cells. And for most of them, we see a reduction in puncta. This is what we would expect for a loss of function. So the protein, if the mutation is detrimental, the protein will not be formed or will have like a truncated version that is non-functional, and we'll have a decrease of the puncta. But except for the splice site here, where we actually almost see like an increase in puncta, suggesting that this might be a gain of function. But all the other mutations behave as we expected for a loss of function, which I believe is most of the mutations in this syndrome. So then we choose one of them, the nonsense mutation, to actually see if our AAV9 is able to rescue the number of synapses. So what you are seeing here is a slice of the organoid. So we slice the organoid in many sections, and then we quantify how many synapses, how many PSD puncta are there, to see if we can actually see like an increase after we add the correct version of the gene. So we have a control cell. This is a normal cell. We have the patient cells, and then we have the patient cells treated with the AAV9. And we use like an IMERIS, which is like an automated, unbiased quantification to actually tell us what is the amount of puncta that we see. And so this is the great news. So what we see is actually the return of the number of puncta, very similar to the control cells, increasing the number of synapses compared to the mutation, the mutant cells without the treatment with AAV. So here is just the PSD95 puncta alone, and here is the PSD95 puncta normalized by the volume of MAP2, which is the volume of the neuronal process, because we want to know if the density of synapses is also corrected, and that's what this graphic is showing here. So this is all good news. I mean, it actually show us that, again, I mean, if we place the correct version of the gene into the mutant cells, we are able to restore the number of puncta, the number of synapses in the process of those neurons. So what are our conclusions and future directions here? Well, first of all, I mean, if you have a brain organoid derived from a patient with loss of function in the DLG4 gene, most likely you're going to have a decrease in synapses in your glutamatergic neurons. So that's great news. We have a phenotype, we have something, and because we reproduce that in many mutations, we are confident that this is real. So most likely in the brain of the patients, they probably have a lower number of synapses as well. So then using the AAV construct, we can rescue the number of puncta from the brain organoids derived at least from one of the patients. The AAV that is clinically great might offer an opportunity to move this idea, this strategy as a treatment, as a gene therapy approach for DLG4 mutations. So what are the next steps? And I've been doing this for several other conditions. So I'm kind of used to know what we have, how we have to think about that. How do we translate what we have in the lab into into clinical trials? And so first of all, I mean, we are validating this in a second patient. We need to test the delivery route of this AAV. Should we inject directly into the brain? Should we inject IV? Should it goes to IV? What are the exactly doses? So is there any toxicity related to this vector? So these are all things that we need to solve before we actually start putting that into patients. And once we have all this data, we prepare a package and we talk to the FDA. Actually, even before the package is ready, we should start the conversation with the FDA, alerting them that we have this therapeutic opportunity and it might translate into a clinical trial. So they become aware and we start thinking about what are the clinical symptoms that we want to rescue. That's why the natural history should comes along with the thinking of a potential therapy. So when we talk to the FDA, we're kind of ready to tell them what we expect to happen if we restore the function of DLG4 in the brain. So these are the next step that we all should be thinking about. All right, so I will stop here by thanking the opportunity to work with you and with the foundation support. I always like to acknowledge the patients who donate the cells. I have a kid with a rare syndrome as well. So I totally understand the burden that it is to collaborate with labs, to donate cells, to have skin biopsies. So thank you very much for that. And most of all, thank you, Luisa, who is the hands of these experiments. Without someone who is specialized.
Hands and brains.
Yeah, thank you.
Great. Dr. Muotri, it's Alec McKenzie, the stumbling moderator here. I think we're going to ask you to hang around and see if there's any questions. Very quickly, there's all sorts of questions. I don't know where the microphones are. So if you guys want to bring them up, and while they're bringing up the microphone, if you did a mouse organoid instead of a human organoid, would things move faster as far as development goes?
A mouse organoid would be much faster. We just don't do it because we have the mouse brain.
Right. Okay, that makes sense. Okay.
Dr. Muotri, I saw that you rescued the phenotype in the nonsense line, but how confident are you that if you rescued it in the nonsense line, that it would work on all loss of functions? Like, should we be worried that somebody else has a missense or a splice site? Are you confident that if that patient thinks that they're a loss of function, that the AAV9 is going to work?
Yeah, we should be. This is my experience with other conditions. Most of loss of function should benefit from this type of treatment. Yeah, we're quite confident that if it is a loss of function, the AAV should help to rescue.
The tricky part comes in the gain of function where there's a dominant negative effect. And
That's correct.
The question is whether or not addition of normal PSD95 when there's a dominant negative will improve. And that's where preclinical models such as that we have to test that before we actually put it into patients will be so critical.
But on that point,
Yep.
Are there other disease types where with a dominant negative effect, because arguably the mutant protein, mutated protein prevents the effective utilization of the good protein.
Yes.
But by inserting
more of the
AAV, you're producing more of the good protein that can develop.
You would think notionally, you think you're
connections.
swamping the bad stuff by giving extra good stuff, and so that theoretically yes, but empirically we don't really know. But to that point, we're sort of 10 years into the gene therapy era, and we should start looking at other disorders where they're dominant negatives and see what they've found out. Tim, excellent point. Yes, so go ahead, sorry. Yes.
Dr. Muotri, just a organoid-related question. So I saw that you showed there are GABAergic and glutamatergic neurons in your organoids. I believe Lucia said these are cortical organoids. So where do you think the GABAergic neurons are coming from? Are there NKX2-1 positive GABAergic progenitors, or are they coming from the PAX6 glutamatergic progenitors?
Yeah, most likely from the PAX6 glutamatergic progenitors. That's why we call a semi-guided, because we give the progenitor cells a little bit of freedom to become GABAergic neurons as well. Yeah.
Okay, thank you.
Has anybody done the creepy experiment of mixing cardiac organoids or endothelial organoids with brain organoids to see if you can actually get some vascularization, or is that too weird?
No, people have done that. So you can mix endothelial cells, but they self-organize into pieces of vases.
Okay.
That doesn't help. I mean, we don't see like a fully perfusible system in the organoid. So that's the gold standard. Yeah, that's where we are all after.
Gotcha. Okay, one more.
Dr. Muotri, so you said that you're confident that you use the AAV9 vector that has been FDA approved, so we just swapped out our gene for one that's already approved. So is it fair to say that we wouldn't have to do preclinical trials in mouse models or large animal models, and we could straight go to the FDA for, you know, IND like approval after? Can we show your data and go, yes, we're ready?
No, you need the toxicology at least.
Right.
Toxicity, because I mean, we don't know if there is anything wrong with these vectors. So at least the toxicology, I'm imagining that the FDA will ask, and I would like to do it before even thinking about placing that in humans.
And what does the toxicology include?
Most likely some kind of large animal, monkeys or pigs, looking if the vector are damaging the cells, if you have cell death, if you activate the immune system, things like that.
And it's Alec McKenzie again. The other cloud on the horizon with AAV in larger patients is that liver failure is being seen with a certain number of them. And so that's something that we need to keep a close eye on as the field matures. I think there may be ways of identifying those at risk with adenovirus infections or something, but nonetheless, that's something to be cognizant of as well, I'd say, yeah.
Yeah, just to complement the reason why we choose like a more clinical grade vector is because if we start adding other pieces in the vector, of course the FDA will ask you what is the security level of all these pieces. So since we have none except for the gene, I think it lowered the bar. I mean, that's why I think the toxicity is going to be probably the only new piece that we need.
Our next speaker has a comment here. Rami?
Yeah. Hi, Alysson. This is Rami Akilan. Wondering if you measured or assessed the functional synapses. Did you look at the multi-electrode arrays and looked at some networking, oscillations, and synchronization?
Yeah, so I think Louisa is doing that right now.
No, she's here at the conference, actually. Well, okay. Your Rett syndrome thing is amazing. Are you just turning the MECP gene back on, or what does that drug do?
The drug activates one of the pathways that is decreased in Rett syndrome cells. So IGF helps actually to produce synaptogenesis. It's not as clean as a gene therapy, but it does the job. So we're not proposing like a cure as a potential gene therapy would do. It should help I mean, it passed the clinical trial, so it's helping the patients.
Wonderful. Okay, congratulations. Sabrina?
Yeah, I have a question from the chat.
Microphone?
Yeah.
Sorry. We'll get 6 microphones next.
Okay, this is about clinical trials. Would the clinical trials you envision be aimed at a specific age group or a range of ages? And I'm just going to add on, I think that the Rett one is with older, with adults as well, right? Yeah,
that's a dialogue with the FDA. So it will be based on the natural history of the condition, the availability of patients, things like that. So that's how they take all these factors in consideration to help establish the first clinical trial. And once you establish either with adults or children, then you expand on trial 2 and trial 3. But the initial population, it's a dialogue. And the foundations will have a lot to say about the ages as well.
The first time it was ever tried in spinal muscular atrophy, they started with infants in the first month of life, and it turned out to be, in retrospect, it's a fairly safe demographic to give AAV, and historically speaking. But you're right, absolutely, Alysson. It'll be a dialogue going forward, I would think. Okay. Okay. I just got to stop talking. Okay.
And this may be my ignorance, and I apologize, but a few years ago, 3-4 years ago, AAV, the big concern was risk factors in that. With the advent of AI, are we seeing actual significant reduction in the risk of AAV?
People are using AI to find the new serotypes or recombinant serotypes that might be less toxic to the liver or might reach better the brain. But these are not in clinics yet. So that's for the future.
Yeah, great question. They're doing biopanning studies where they put into large non-human primates AAV with a whole bunch of different surface molecules and identifying one that showed different sort of what we call tropism. Yeah, so exactly right. I'm doing too much talking as a moderator. Sorry. Okay, Dr. Muotri, thanks very much for an excellent talk. It really energized people after lunch, so thank you.
Thank you.